Okay, thank you, Marianne, for this kind introduction. Thank you to you and also to Cameron for organizing these webinars, which keep us connected in this special period. And thank you for giving me the opportunity to talk today. So I don't pretend to have the talent of Gabriel Garcia Marquez, and I apologize to the lovers of this famous writer for diverting one of his titles to contextualize a talk which would be dedicated to molecular surveillance of NTDs. But I believe, like most of you, that in the time of Corona and probably afterwards, research and healthcare on NTDs is and will be strongly impacted. In the time of Corona, the humanity has never been so well informed and concerned about infectious disease. Everybody knows about the virus. Everybody is used now to terms like epidemiology, control, PCR, serology, DNA sequencing, and strains. Everybody realizes the importance of research and good health systems, and this is an unprecedented achievement in education to health, and I mean it. I'm working in an institution aiming at the advancement and spread of sciences that contribute to tropical medicine and public health in developing countries, inclusive of their global dimension. Thus, for us, Corona is a key priority in Belgium, but also overseas, but it is also a priority among others. And as molecular parasitologists, I might sometimes currently feel somehow useless for health in my country, but I'm still concerned by neglect of parasitic diseases at global level. Because at that level for parasitic diseases, a minority knows that these parasites kill, incapacitate, and impoverish at the other side of the world. Means engaged in the search for new diagnostics, drugs or vaccines are too low, and few bother about flattening of the curves in control program of NTDs. And I don't think I need to convince this audience about it. And yet, we may have reasons to be positive, as NTD control programs are achieving their objectives. If we look, for instance, at the three main diseases due to trypanosomatids, anthroponotic visceral leishmaniasis, AVL, on which I will focus my talk today, sleeping sickness, and Chagas disease, we see significant drop of cases. But despite these successes, these programs remain vulnerable to decrease and shift in political attention and priority, and this is quite likely in the current Corona crisis context. These programs are also confronted to biological and operational challenges. And here again, Corona might play an important role. For instance, by reactivating, maybe in the future, asymptomatic parasitic infections in weakened individuals, or by destabilizing health systems in NTDs endemic countries. Accordingly, I'm afraid that new outbreaks of NTDs are likely, and they could impact current elimination programs if care is not taken. In his review on AVL, Suman Rijal was identifying 4 essential actions in post-elimination phase: developing improved diagnostic tools, new drugs, adapting vector control measures, making case detection and epidemiological surveillance. And in my talk, I will broaden the last point and pay a particular attention to molecular surveillance through pathogen genotyping, which is something which is well embedded in diseases like TB, foodborne disease, or COVID, but which is highly neglected in NTDs. And yet molecular surveillance is susceptible to answer several outstanding questions of public relevance. I listed 6 here. Following the evolution of epidemics in time and space, undertaking outbreak studies and identify the sources of these, characterizing new transmission cycles, detecting new variants of the pathogens, defining genetic markers of relevant traits like drug resistance, for instance, and assessing recombination. And these questions could be answered for parasites by targeted genotyping methods. I mean methods that target a set of predefined molecular features, like multilocus microsatellite typing. But these methods, these targeted methods, suffer several limitations. They are far from being standardized. They need multiple assays. They identify known mutations and don't allow discovering new ones. And their discriminatory power depends on the evolutionary history of the pathogen in question. And on this slide here, you see a phylogenetic tree of the Leishmania donovani complex, where you see that most of the isolates can be distinguished, except those which are here in this red line, which are isolates from the Indian subcontinent, which are totally identical with the technique which was used here. It's impossible to do any molecular epidemiology or molecular surveillance in such conditions. In current post-genomic era, I will try to convince you that whole genome sequencing is a powerful method for molecular surveillance, and for this, I will use our work on AVL in the Indian subcontinent as model and show how a single method allows answering the 6 outstanding questions I was mentioning. First, following the evolution of epidemics in time and space. In 2 research projects that we undertook in the years 2000, we collected about 200 clinical isolates of Leishmania donovani from the epicenter of the epidemics of AVL in the Indian subcontinent. There were parasites from India, from Nepal, and from Bangladesh. These isolates correspond to the red line I was showing you in the MLST-based tree. And here below, you see the result you get when you analyze the same parasite with whole genome sequencing. And you can see easily that we can distinguish each isolate and that we could even classify them in main genetic groups called IC2 for up to IC10. Using the same tree, we then timed the main events underlying the evolution of this whole population. And we first found that its origin was around 1850, and this is based on the genetic data, and this corresponded to the first reports of an epidemic of AVL in the Indian subcontinent in Assam. The second event is shown in this slide, where you can see that the Indo-Nepalese population, which is here, oops, sorry, I have to come back. Just which is here. This group is isolated from Bangladesh. The origin of this Indo-Nepalese population was timed around 1900, which again corresponded to the literature because that was the first reported epidemics of AVL in Bihar, in India. And then a third event was the spreading and diversification of these different IC groups, which is estimated around 1960, just after a major anti-malaria DDT campaign. And it highlights the bottleneck that was caused by the campaign and that was followed by a series of outbreaks, resulting in these different groups. And it is during that last period that antimonial treatment failure dramatically increased in the Indian subcontinent, causing the loss of these drugs for chemotherapy in that region. The whole genome sequencing data was highly relevant for describing the outbreaks of the past and they constitute an essential reference for the future. Indeed, in case of new outbreaks of AVL arising now in the post-elimination phase, we could easily define a source, either a genotype originating from an existing group like this one, or a genotype coming from abroad, or a new and yet undetected genotype. This is very easy with the performance of the whole genome sequencing. This brings me naturally to the third question, which is the characterization of new transmission cycles. During the study I presented to you, most isolates were coming from the lowlands, which are here in the Ganges plain, which is the classical biotope of AVL. But we also got some access to some parasites, some VL parasites from a new focus on autochthonous transmission in the highlands in Nepal. And as you can see on this zoom of a phylogenetic tree, these parasites from the highlands, which are here in blue, are quite different from the parasites we find in the lowlands. And this new group of Eldonovani coming from the highlands were called Yeti parasites. That was the nickname we gave them in the name. It didn't please the editor of the paper, so we had to call them ISC1 in the literature. But these Yeti parasites have a very distinct history from the parasites from the lowlands, and they were possibly imported from another country, and they could even be from East Africa. The same genomic sequence data allowed to characterize deeply these new variants and find new features in them. And briefly, we found that these Yeti parasites showed strong genomic signatures of different virulence and different susceptibility to drugs. And they also showed to be very well adapted to Phlebotomus argentipes, which is the vector of Leishmania donovani in the lowlands. Thus, in short, our results show that we have here a new and very different variant of Leishmania donovani that could easily spread over the whole lowlands if care is not taken. The analysis of genome sequencing data gave us also a detailed insight on the history of drug resistance in that population. First, it showed that the road to resistance was long and occurred in several steps. First, there was a pre-adaptation to antimonials, which was already present in 1850 when the population emerged, the lowland population emerged. And this adaptation brought a relative tolerance to antimonials. And what is very interesting is that this pre-adaptation occurred years before the antimonials were introduced. And this is a big mystery, and one of the possible explanations is that these parasites in 1850 were exposed, naturally exposed to other compounds like arsenic. Thus that was the first step in this road to resistance. Then the second step that we identified, at least in our data, was in this population here, there was, this is a genetic group which emerged and which is characterized by a 2-nucleotide indel in AQP1. AQP1 is a major transporter of antimonials, and this indel renders this AQP1 non-functional, and this decreases the susceptibility of the parasites to antimonial. And then later on, independent mutations emerged here and there in other genetic groups, and they also decreased the functionality of AQP1, but these mutations didn't spread yet. This brings me to a second concept that we call the many roads to resistance. In other words, several distinct mutations in a given gene can contribute to resistance, like a full deletion of this AQP1 gene, or mutations affecting key residues, or the indel I mentioned you, or other type of events. And you can easily realize that a single PCR assay that we would design to detect one of these mutations would not detect the other ones, which again shows the strong power of whole genome sequencing for the search of known and new biomarkers. And last but not least, the resolution of genome sequencing allows to track recombination between parasites. And this is illustrated here by the color code in panel C. And look at IC5 here. This is predominantly green, which corresponds to a pure population. And you see here another population, which is a mixture of green and other colors. And this is the signature of two genomes that hybridized. Thus, these parasites are hybrids. And it is highly relevant from an epidemiological point of view, as it implies that drug resistance genes could be transferred, for instance, from this parent to these hybrids. And we found that here in this graph, as you see here, the bars represent the percentage of reads containing the mutation in the AQP1, and you see that all the parasites from this group, they have about 100% of the reads, while here in these hybrids they have a variable proportion depending on the genome that has been given to them. In conclusion of this first part of my talk, we saw thus that whole genome sequencing is very powerful. It gives us the possibility to track any strain, even in a very young population like the one of the Indian subcontinent. We could track the past epidemics, and we detect several major waves in 1850, 1900, 1960, and which are histories of bottlenecks and clonal propagation. And as I told you, we have the baseline data to identify next waves, if it would come, hopefully not. We identified a new variant, this so-called Yeti strain, which is functionally different from parasite from the lowlands and well adapted to Phlebotomus argentipes. And we also showed that the technique was very powerful to detect markers of resistance. And we showed that the history of the epidemics in the Indian subcontinent was a long road to drug resistance with various steps. And what was also very important was this concept of many roads to drug resistance, the fact that different mutations can lead to drug resistance. And we, last but not least, we found several signatures of recombination. Thus, whole genome sequencing-based typing seems to be highly recommended for surveillance. I hope I could convince you about this. But the method should still be upgraded. Indeed, during my whole talk so far, you heard me speaking about typing of isolates or strains, thus parasites that were brought in a culture medium and propagated in vitro before DNA analysis. And this is the procedure used so far for all whole genome sequencing studies published on trypanosomatids. But this approach is actually associated to severe sampling and biological biases. First, a large proportion of samples remains inaccessible if we focus on cultured isolates. Indeed, the isolation success of parasites from clinical samples is only around 50%. 90% of the Donovan infections are asymptomatic, meaning that we miss a large proportion of the population diversity because we sample only in clinical patients that present to the health services. And another problem, and this is more a biological bias, is that we showed experimentally that depending on the life stage and on the host of the parasite, the genome could be different. In other words, based on these facts, we thought that it should be important to sequence not anymore cultured isolates, but to sequence directly the parasite in the host tissues. But there are many challenges associated to this, because there are very low amounts of Leishmania DNA, high abundance of host DNA, so impossible to sequence directly. And therefore, we need to develop a method for enrichment of Leishmania DNA. And in the second part of my talk, I will speak about this endeavor. And for this, I borrowed slides that were made by my collaborator, Gosha Domagalska, who's the leader of this project on direct Leishmania genome sequencing in clinical samples. And I will present these slides on her behalf. The first slide presents you the magnitude of the challenge if you want to do direct sequencing. You can see here that the median percentage of Leishmania DNA versus human DNA is very low. It's about 0.02%. Thus, impossible to find a needle in a haystack without an adequate enrichment technology. Among a few alternatives, we choose the Sureselect approach, which is based on the use of probes. There are more than 200,000 probes of Leishmania that cover about 75% of the genome. And these probes are hybridized to a complex mixture of Leishmania and human DNA. And the fragments that hybridize the Leishmania DNA are then fished and sequenced. We first assess the analytical sensitivity of the method with simulated clinical samples, and we found out that a good Leishmania sequence quality could still be obtained with a starting concentration of 0.006%. And the enrichment factor in these conditions was about 500. At lower concentration, there, the quality of the sequence was not okay. Then we went back to Nepal. We were actually lucky because at the time we had isolated the parasites presented in the first part of my talk. We kept bone marrow in the freezer just in case the method would allow direct analysis in these. And time has come for it, and we just applied our enrichment method on these. And the next slide shows you the difference in genome coverage when applying the enrichment method, that is in green, or not. If you don't apply enrichment, this is the coverage you have in orange. And you see clearly the difference. And actually, in 53 out of 62 samples, the sequence quality was good for in-depth genetic analysis. This phylogenetic tree showed that it was possible to attribute the sequenced clinical samples, which are figured here in dots, to the groups defined in previous work. So the topology of the tree was respected, and we did not see new genetic groups appearing in the tree. However, the most striking result is shown on current slide. We had 12 patients where we had the paired bone marrow and the cultivated isolate derived from it. And thus we could sequence the two of them. And to our big surprise, in the 12 cases, the genotype encountered in culture was different from the one observed in clinical samples. And this was likely explained by the existence of a polyclonal infection in the patient with a dominant clone more fit to in vivo condition, and this is the one that appears when we sequence, and probably there are minor clones which are hidden, not detected when you sequence, but when they are put in vitro, these minor clones could become dominant in culture because they are more adapted to it. And you can easily imagine the impact of this, for instance, for the search of biomarkers. And we had a case where the dominant clone in vivo harbored a drug resistance mutation, but this was missing in the in vitro dominant clone. How can you then predict treatment outcome in a patient with a genotyping made on cultured isolate? Just it is clear that molecular surveillance should be done directly on clinical samples. We believe that this study represents a major milestone in genomics and its application for molecular surveillance, at least for trypanosomatics. And we hope it will inspire future work on the following points. Application on other Leishmania, we should also see how the method can be useful for other trypanosomatid diseases, sleeping sickness, Chagas. Another question for research is the choice of tissues for doing this type of research. Of course, we could be tempted to look for the tissues which are easier to collect, where you have a higher parasite load, but we have also to think in terms of the question we want to address. If we want to understand the transmission, if we want to understand pathogenicity, drug resistance, which tissue is the best one? I don't know yet. We should work to increase the analytical sensitivity of the method. You saw that it's so far the sensitivity is about, the detection level is 0.006%. If we want to study, for instance, asymptomatic samples, we will need to increase this sensitivity. And we should work on strategies to sample in less accessible foci. And what would be really relevant is to integrate this type of molecular surveillance platform in a global surveillance platform with clinical and epidemiological data, with bioinformatic tools, and of course for this, stakeholders must be included. So I will stop here. If you're interested in these studies, I can recommend you to read the review that we just published in Trends in Parasitology, and that will be made available through the ISNTD platform. This work was made thanks to an excellent collaboration with the Sanger Institute, especially James Cotton and Matt Berryman, and all the work on the field to collect the samples, these isolates, was impossible without the help of BP Koirala Institute of Health Science in Nepal and Banaras University in Varanasi. If you want to contact us for further details, you see here our respective emails. And if you want to have more information on the further result of our studies, you can follow us on the Twitter account of the laboratory. So I thank you for your attention. And yes, I'm available to answer a question. Thank you.
Thank you very much, Professor Dujardin. That was quite a very detailed and in-depth review of all the possibilities and opportunities that are out there in terms of obviously the acute science, but also integrating this sort of medium to long term into the next steps of public health programs in NTDs. And that's something that WHO has been thinking a lot about, launching a new roadmap very soon for the next steps in the control of NTD. So that definitely somewhere where partnerships in this field will make a huge difference to consolidating the successes that have already been gained. Professor Dujardin, you've got a lot of thank yous here on the chat. Everybody really enjoyed your talk, saying thank you so much for the presentation. And we did have a few questions. So one of the first questions was from Omar Harb, who's asking, was there a correlation between the minor clones and the major clones discovered in the polyclonal infections? And before you answer, if you also want to include at the start of your question which organization you're from or which country, that would be really useful as well.
Okay, I'm not sure I understood well the question about this this correlation between minor and major genotypes. This is still work in progress, because we found only we could really demonstrate this polyclonality of the infections in a few cases, because it's very difficult to detect these minor reads. But what I can tell you is that we made some experiments. We tried to reproduce these experiments experimentally. So we mixed different clones together at different proportion to see to verify at least if in vitro there would be always the same clone that would come out because having a higher fitness. And this was the case. Because there we saw this this reproducibility. In vivo, it is impossible to verify that for the moment. But I think it would be interesting to work on that in the future, to explore more this polyclonality issue and to document its extent. This is also a point that could be interesting to study between the organs, because if there is polyclonality in a given host, it is possible that between different organs, different clones could be dominant because they are more fit to the skin or to the bone marrow, for instance.
Brilliant. We've got another question here, quite a few questions actually, so I'm just taking them slightly in order of them appearing on our system. Dora Johnson, who's typing in from Bangkok, I believe. Hello, Dora. Thank you so much, Professor Dujardin, for this amazing presentation. I have a question. Does the molecular surveillance, can this also be applied for the disease vector, and what could be the relevance of doing that from an entomological viewpoint in leishmaniasis?
I will try to see the question, because I didn't. You mean to the vector? I didn't get the point about the vector.
Yes. So can you apply these techniques, molecular surveillance, to the vector itself?
To the vector itself.
Yeah, the vector itself.
I imagine, yes. I think these are techniques that can be applied to any organism. So I don't see any problem for that. I don't know who has done that yet in the world of entomology, but that should not be a problem.
Brilliant. And could you see this as having, what could be some of the connections with entomological programs or vector control programs from a viewpoint of leishmaniasis? That was the second part of Dora's question.
You mean just to, how would a molecular surveillance of the sandfly, for instance, be relevant for the control? Yeah, I'm not an expert in entomology, but I think with a resolutive tool, it would be possible to identify in a very precise way, for instance, the gene flow in the population of insects, and this could give information. For instance, if there is a control activities with spraying of the insects, and there are insects coming back, this technique would allow, for instance, to see if these insects are the same as the ones which were previously. So meaning that maybe the spraying was not perfect, or that there has been a reinvasion from somewhere else. And for that, you need very resolutive power. Maybe it could be possible with other method than whole genome sequencing, because if my message and my plea for molecular sequencing was especially in the context of the parasite of the Indian subcontinent, because there we cannot do molecular epidemiology without whole genome sequencing, but it's sometimes in other populations with simpler technique like MLST or MLMT, it is possible.
Brilliant. Slightly different question from Jérôme Estaquier about the two patients that in which you observed higher diversity between parasite and in vitro. Was the initial amount of parasite similar or higher, or was it the time of culture isolation that created parasite diversity?
Thus in these 12 cases, I don't have any clue or available now here in my hands on the parasite load at the beginning. The isolates were analyzed. Yes, they were analyzed about 20 passages after the moment of isolation, and we know that very quickly the parasites can change. It's not that the parasite changes, yet you have probably this polyclonal population at the beginning, and then you go, isolating is going through a huge bottleneck. Only a few cells will survive, and these are the cells which are fit to the culture medium, and these will then propagate. Thus, it's not that the isolation creates mutation and makes the parasite changing. No, there is a heterogeneous population, a polyclonal population, that's at least what I think, in the patient. And then during the bottleneck of the isolation, you have only a few and different ones which will grow.
Brilliant, thank you. Just a little hello there from Paul Denny, who says, thanks, Jean-Claude, much appreciated diversion. And following from that, a question from a colleague at ITM Antwerp, Gert van den Aura. What is your vision on complementing parasite molecular surveillance with host genetics?
I'm very happy that Gert didn't come with more difficult questions, because generally he asks, and how is this useful for solving the problem, etc. Thank you, Gert. I think it would be interesting to do this type of studies, and the approach we are using allows this, because we have the Leishmania DNA and the human DNA together. I can directly reassure you that we didn't touch the human DNA in these studies, because there was not ethical agreement for doing this, and thus the human sequence have been automatically deleted. But indeed, it would be it's a technique which would allow this type of studies. But of course, the sample design and the sample size should be probably much higher to do this.
Great. And Catherine Brown is asking, first of all, saying thank you for the excellent presentation. Do you have any WGS data of isolates from asymptomatic carriers? And if so, any suggestion of attenuation?
The huge reservoir of parasite which is there is different or exactly the same that the one we see in the patients. And we are working on different alternative ideas to increase the sensitivity of the method to allow to genotype these very low amounts of parasite. There are different possibilities. We could work with selective whole genome amplification. That's the technique that has been used in Plasmodium. Another technique that could be used is to take the opportunity of the difference in methylation between the Leishmania DNA, which is very poorly, there is very low methylation, and human DNA where there is higher methylation. And it is possible on the base of this to get rid of a big proportion of human DNA. So by combining Sureselect, for instance, with this demethylation, as we call it, we could get a higher sensitivity, but this has to be tested. And we look for collaborators who would be interested to join us in this type of research.
Absolutely, a call for partnerships there. And by the way, Gert is saying don't tempt him to ask a harder question. Moving on here, a question from Kamaluddin Benalal. Thank you for the presentation. Could you give a bit more detail about the relationship between malaria control and the emergence of drug resistance in Leishmania?
That's a very interesting question. Just in one of the slides, I showed you that in 1960 there was a new series of outbreaks with a spreading of these different ISC population. So what happened probably at that moment is that the malaria control, which was a DDT spraying based control among others, eliminated, of course, a big proportion of mosquitoes, but also sandflies. And does this cause indirectly a bottleneck in the transmission of Leishmania donovani in the Indian subcontinent? And then when that was stopped or decreased, then the insects, including the sandflies, could spread again, and then there was an outbreak again. No, that's the explanation why the malaria control can have an impact on the outbreaks of leishmaniasis. The relation between the malaria control and the emerging of drug resistance, I don't see a direct link between the two. If there is something, it's simply because the malaria control allowed the parasite, the epidemics to spread again, that there was a big biomass of parasites, and these parasites, one of them solution to the antimonial, and then this was spread. That was this mutation in the AQP1.
Great, thank you very much. We had here, perhaps we could give a hand to Vishnu Joshi, who is from Nepal and doing a PhD in Norway. Do you think dual RNA sequencing will help to know better insights in host-parasite interactions?
Yes, well, there we are, of course, far from molecular surveillance. It's more in basic studies to understand this important issue of host-parasite interactions. Yes, I think studying the RNA of the parasite gives you already, you are more close to the function and to functional aspect than if you use genomic data. What has been done by others, and which is probably the most interesting to do, is to combine studies by RNA of the parasite and the host cells. Thus, it's what is called co-transcriptomic studies, and there you can study the functions of the parasite, and in the context of the function of the macrophage, for instance, if you do this type of study. So that would be definitively something very interesting. And I would even go further now with the revolution of single cell omics. I would go single cell RNA-seq for doing this type of studies, because with that you can really get a much better perception of these interactions.
Brilliant. Professor Dujardin, we've had the pleasure of having you for a good 45 minutes now, so I was just wondering whether, yeah, I see you're quenching your thirst there, whether you might still have a few minutes for one or two more questions, if that's okay with you. I'm quite happy to carry on, but obviously, your time is very much appreciated.
If it is not a question from Gert van der Aura, it's okay.
So far, so good. He hasn't added anything, but don't tempt him. I've got here actually a question from Helena Uliartha from Indonesia, Ministry of Health, who's saying, thank you, Dr. Jean, very interesting. It's like having my biomolecular classes again. I agree, WGS is important in an elimination setting in Indian subcontinent, for instance. WGS may be difficult because the infections are getting lower and the drug resistance also is an issue. What do you think about a vaccine?
About a vaccine? I'm not at all an expert in vaccinology. The only thing I would say, and this is really my personal opinion, I insist on that, having studied these parasites for some years, I realized they are really very complex. And they are very complex, and they are also very flexible. They have multiple tricks, for instance, to escape the attack of the drugs. And I know from experience that vaccine against eukaryotic parasites, this is really a huge challenge. So I think it's an important avenue. It has to be explored, and there are already some vaccine candidates for dogs, for instance. I think we are still far from having a vaccine for humans, but of course it would be nice if it was discovered. But I think the road will be long also there to develop a vaccine against leishmania or other trypanosomatids.
Okay, well, I think Gert's planning something, by the way, but in the meantime, let's take a question from Adugna Bera Hirpa. Hi, I'm Adugna from Ethiopia. My question is in the context of visceral leishmaniasis elimination, and particularly the role of molecular surveillance in settings where there are individuals with asymptomatic infections, and how can we deal with these asymptomatic infections? I mean, are these cases treated for visceral leishmaniasis?
As far as I know, asymptomatic individuals are not treated for two reasons. First, they are not systematically diagnosed. Maybe in some studies you can have an idea of who is asymptomatic, but there is no systematic diagnostics of asymptomatic individuals. I even don't know if there are methods which are validated for that. Secondly, the treatment is toxic, expensive, and for the moment, the treatment is only given for patients, for people who show symptoms. If in the future another type of treatment, cheap, less toxic could be used, we could consider that. But this is still very risky because putting more the population of the parasites under pressure that could also be a way to select resistance or tolerance to the drug. So, yeah, that would be my humble answer to this question.
Great. And rather than putting you on the spot, Gert has actually come to give you a little hand here, just going back to the vaccine question that we just heard about. Gert says, what I recall from a workshop on vaccines in Iran some years ago with Institute Pasteur, a vaccine would be cost effective only if developed, for all Leishmania species. And that would be quite a challenge. But again, he says, like Jean-Claude, I'm not an expert on this. I just thought.
Thank you for this comment.
Yeah. Okay, perhaps coming on to our final two questions. Another question from Kamaluddin Benalal. Could sandflies have a role in resistance emergence?
That's a very interesting question. I have to think a few seconds. Of course, they could have a role in the spreading of the resistance once, for instance, if the resistant parasite is more or less fit in the insect, because if for any reason the resistant parasite is not fit in the sandfly, yes, it will have difficulty to be transmitted. But that's for the spreading. In terms of the emergence, I had a suggestion a few weeks ago made by a colleague that was at the BSP meeting in Granada, because I was speaking about pre-adaptation to drug that was existing in Leishmania. And he told me that it would be interesting to have a look at the biotope which is in the gut of the sandfly, because there are bacteria, there can be fungi, and he was telling me that there are metabolites there which could have an action to on the parasite, and that the parasite would have to adapt to these natural metabolites of the microbiota, and this could maybe then pre-adapt these parasites to become resistant the day they encounter a molecule which share similarity with the metabolites that they have been exposed in the sunfly. It's totally speculative at this stage, but I found it very interesting, this comment of of my colleague, and it would be interesting to make this type of studies. Again, it's more basic studies on the interaction of the parasite with metabolites of the fauna which is present in the gut of the sunfly.
Brilliant. And so, very final question. I think we are coming up to the end of today's webinar. A question from Emmanuel Nandi. Thanks, Professor, for the wonderful presentation. Please, are there information on biomarkers to detect the mutant species of this organism?
To detect the mutant species?
Yep, sorry, I just lost. Probably, yeah, mutated species of.
It depends what biomarkers of what we are searching. It can be biomarkers of resistance, could be biomarkers of virulence or pathogenicity. One of my final point would be that if for instance somebody wants to find markers in the genome for higher pathogenicity in the patient, and this is something I have been searching for years before I was working on drug resistance, and I didn't find anything. And of course, we know that pathogenicity could be due to the host, the host factor, and not only the parasite. But I think that one of the reasons why most of the studies didn't find biomarkers, for instance, for pathogenicity, is that we were working with isolates. And as you have seen, the isolates are not necessarily representative of the parasite which is found in the tissues of the patient. And thus, if we want to do clinical studies and making a parallel with it between a parasite feature and the clinical study, I think we need to do these studies directly in the host tissues and not on isolates. And I think that many of the studies that have been done in the past have to be redone now in this way.
Brilliant. And so, would you like to conclude with a question from Gert?
Why not?
Why not, huh? It would be a nice symbol, I think. Does resistance in promastigotes also mean resistance in amastigotes?
Yeah, these are two life stages which are very different. We have examples of drugs which are more susceptible. Sorry. We have life stages which are more susceptible to a drug. That is common. Sometimes promastigotes are very susceptible, or the amastigote. So what I would like to say to finish on that is that drug resistance is one issue, but there are other things which have to be considered when we think in terms of the susceptibility to the treatment. That's the quiescence. Because, and this is a topic, a field which is emerging now in the recent years in the Leishmania community, but also in Trypanosoma cruzi, is the fact that parasites can start to sleep and reduce their metabolism when they are exposed to stress, and this can bring them a tolerance to the drug without developing a drug resistance. And this is something which should be explored in the next years, because it could explain a lot of things. Because we have seen in our own experience that the drug resistance phenotype which is a phenotype that you measure in vitro with isolates again, has a very poor predictive value of the treatment outcome. In our experience, if we had a Leishmania resistant or sensitive to a given drug, we could not predict very well the outcome it would be in the patient. And one of the reasons again could be that the treatment outcome in the patient is due to hemastigotes which are present in the patient, and the tool we use to measure the in vitro resistance is something we do on promastigotes or hemastigotes, but isolated from the patient. And this could bring a lot of discrepancies. Because I think there is a lot to revisit in terms of the link between resistance and treatment outcome, and an essential aspect that may not be forgotten, and that will be more and more split in the future. That is this issue of quiescence.
Very thorough reply there. Thank you very much. And so this brings us towards the end of today's webinar. Professor Dujardin, thank you so much. This was a really important time and an important topic to put on the map. Thank you as well to all our attendees. Thank you, Helena, Kamal, Gert, Jérôme, Huguette, who tuned in from Cameroon. We have Dieter from Princeton, Ariel from Brazil, thank you, and hello, and Isa from Nigeria. I can't name everybody, but a very big thank you. Judith, Dora, thanks very much, and we hope to see you very, very soon. I hope that in this time of COVID, in the time of Corona, that global awareness of the role of surveillance, and therefore the possibilities that are out there in terms of the technology available, it will really be put on the map, and that this will mean something very new, a new path going forward for NTD diagnostics. So for that, thank you, Professor, for putting it on the map. I just wanted to remind everyone, a few people were asking on the chat that this webinar has been recorded, and if you want to go back and listen to it again, or perhaps share it within your network or your friends or co-authors, we'll send the link around it should be ready in about 2-3 hours. Please also do consider joining some of our future meetings. We're having them on Tuesdays and Thursdays at the moment, and they last anywhere between half an hour and up to an hour as today, and I will just try and pop up the forthcoming schedule. So here we go. So next week on Tuesday, we're really thrilled to actually be able to welcome Dr. Mwele Malachela from the World Health Organization, head of the Department of NTD Control. And following that, a panel, actually, we'll try and bring in a number of speakers talking about hand washing. And in the week after that, on the Thursday, there'll also be a DNDi panel looking at R&D in neglected tropical diseases. And on the right-hand side, you can see which videos are currently will be available for replay. We've got from the clinician, Dr. Gabriel Yan, we've got a look at the threat of dengue in the time of COVID, and his experiences as a clinician and some of the patients he's seen. We've also had a talk by Dr. Frederick Seelig looking at vector control and the possibility to partner on the Global Vector Hub. And finally, after looking at all this very hard science, we do invite you in the week of April 21st. We're going to have a session a little bit more relaxed and looking at some of the absolutely wonderful creative approaches to messaging and communication about infectious and tropical diseases, and looking at films, apps, maybe a few games that are out there, and all that is kind of a very pared down and summarized session based on our annual ISNTD festival, where we go all out and bring in all sorts of partners working in a lot of different creative and communication approaches. So we look forward to keep connecting with you and to keep connecting everyone with each other. Just trying to get back to my screen. There we go. So on that note, hope to see you all very, very soon. Another big thank you, Professor Dujardin. A round of applause if everyone could do that from home. And we hope to see you very, very soon. And thanks again, Professor.
Thank you. Bye.
Take care. Bye-bye, everyone. Keep safe.
Comfy.
Yeah. Keep comfy, keep safe. Take care. Bye.
Bye.